Hi everyone. Today we are going to discuss about cholesterol metabolism. So what do you know about cholesterol? Cholesterol is a steroid compound made up of 27 carbons. It has a cyclopentanoperhydrophenanthrene ring. There is a OH in the third position and you have double bond between 5 and 6 carbons. It is an amphipathic lipid. And in addition, we saw certain special products derived from cholesterol, which is vitamin D, bile acids, and steroid hormones, right? So with this short recap of what you know about cholesterol, let us see today about the synthesis of cholesterol. Cholesterol synthesis is a very commonly asked essay question in university examinations, and also there are many potential spaces where MCQs can be tagged from this particular topic. So let us see where this cholesterol synthesis occurs. The site of synthesis of cholesterol is liver, intestine, and almost all the nucleated cells. And which organelle does this synthesis occur? It is going to be partly in the endoplasmic reticulum and partly in the cytoplasm of all these organs. And the starting substrate from which cholesterol is synthesized is a 2-carbon compound, which is acetyl-CoA. So from a 2-carbon compound, you're going to synthesize a mega compound of 27 carbons. Then the key enzyme for this particular synthesis is HMG-CoA reductase. Each one of these is a potential MCQ question for you. Now let us see the steps of synthesis. The synthesis has many steps. So in order to just summarize it, we have just compiled it into 5 major steps. First one is synthesis of mevalonate from acetyl-CoA. Then you have the formation of isoprene units from mevalonate. Now you condense 6 isoprene units to form a compound known as squalene. Now cyclizing squalene to form lanosterol is the next step, and finally you form cholesterol from lanosterol is the next step. So these are the 5 major steps. Now let us see into each individual step among these major steps. The first one, synthesis of mevalonate from acetyl-CoA. This is a very important step because it contains the rate limiting enzyme. Okay. So we have started from acetyl-CoA, right? You combine 2 molecules of acetyl-CoA, 2 carbon, 2 carbon. So a 4-carbon compound is formed, and that 4-carbon compound is called acetoacetyl-CoA. This is by the enzyme thiolase. Then you add 1 more acetyl-CoA. So now 4 plus 2, it is a 6-carbon compound, and that compound is HMG-CoA. The enzyme is HMG-CoA synthase. So up to here, the synthesis of ketone bodies and synthesis of cholesterol is following the same sequence of steps. After this, cholesterol synthesis goes into the regulatory step, that is a HMG-CoA reductase step. So through HMG-CoA reductase, HMG-CoA is converted into a compound which is mevalonate. This is the first set of steps in the synthesis of cholesterol. You have synthesized mevalonate. Now, since we told HMG-CoA reductase is a key enzyme, now let us discuss about the significance of this particular enzyme. It is a regulatory step of cholesterol synthesis, and it is also the site of action of the most common group of hypolipidemic drugs, that is statins. Statins are used by almost every individual with dyslipidemia, and more commonly individuals those who are at risk for dyslipidemia, such as diabetic individuals. So statins are a group of drugs which acts by inhibiting the key enzyme of cholesterol synthesis, that is HMG-CoA reductase. This is a very commonly asked MCQ question. Now let us see how this HMG-CoA reductase is going to regulate your cholesterol synthesis. So regulation means, see, whenever there is a requirement for cholesterol, it needs to be synthesized. Whenever there is no requirement, it should be stopped, right? So how this check occurs, whether it has to occur or do not occur, is included under regulation. So HMG-CoA reductase is the regulatory step. How or what are all the factors which can regulate this particular enzyme? The first one, you all know, products. When you have the products in excess, the reaction will not occur. So the product of that particular enzyme is mevalonate, and the end product of whole metabolism is cholesterol. So when mevalonate and cholesterol are in excess, this particular reaction or the synthetic pathway will not occur. The next one is covalent modification. So attachment of a phosphate group, a phosphoryl group to a particular enzyme can either activate it or deactivate it, and that is a form of regulation of enzyme activity. Here, HMG-CoA reductase is active in the dephosphorylated state. This is also a very commonly asked MCQ question. Then we have various hormones. You all know insulin is an anabolic hormone, right? So any synthetic pathway, it will increase. So insulin increases cholesterol synthesis and glucagon is a catabolic hormone. So insulin will not increase this cholesterol synthesis pathway. And then you have a compound which is SREBP, sterol regulatory element binding protein. And this has a major role in the regulation of cholesterol metabolism. Let us see the role of SREBP in regulation of cholesterol synthesis. So here if you see, you have the endoplasmic reticulum membrane and this side the cytosol and the lumen, right? So whenever you have excess of sterol compounds, here oxysterol and or various sterols, they remain bound to the insulin induced gene, that is INSIG gene, and this remains attached to a protein which is SREBP cleavage activator protein. This cleavage activator protein holds on tight to this SREBP and does not let it out. So cholesterol synthesis will not take place when there is high levels of sterol. Whereas if there is low levels of sterol in the endoplasmic reticulum, what happens is ubiquitin will go and target this INSIG for degradation. So once this INSIG goes for degradation, now the SCAP protein is left alone, right? This secretory proteins will escort the SCAP and the SREBP into the Golgi apparatus. So inside the Golgi, we have two different proteases and that will cleave or set free the regulatory domain of SREBP. Now, this SREBP regulatory domain will enter into the nucleus where the genes for the enzymes of cholesterol synthesis are located, and that will lead to increased transcription of those particular enzyme genes. This will lead to increase in the cholesterol synthesis. So, this is how the protein SREBP mediates the regulation of cholesterol synthesis. So whenever the levels are very high, SREBP stays along with SCAP protein and it never moves out of the endoplasmic reticulum. Whereas when the levels of cholesterol or sterol levels are low, then this particular SREBP gets cleaved and the regulatory domain will go inside the nucleus and increases the transcription of the various enzymes required for cholesterol synthesis. Now coming to the next major group, that is after mevalonate synthesis, you are going to form the isoprenoid units. So what is this isoprenoid units? Isoprene units means 5 carbon compounds. Okay, so mevalonate is a 6 carbon compound. You are going to form a 5 carbon compound from that. So what are the various steps in that particular step? So initially you need to add 3 phosphate groups to this mevalonate one by one. So to add phosphate you need a kinase. So you add 3 kinases and highly activate this mevalonate. Then after that you remove a carbon to form the 5 carbon compound, right? So the first is mevalonate kinase which forms mevalonate phosphate, then phosphomevalonate kinase which forms mevalonate diphosphate, and diphosphomevalonate kinase which forms mevalonate phosphodiphosphate. Okay, so three kinases, so 3 phosphates are being added to mevalonate, and finally a decarboxylase, remove one carbon. So you have a 5 carbon compound which is isopentenyl diphosphate. This can also get isomerized to a compound dimethyl allyl diphosphate. So both of these are 5 carbon compounds and these are the isoprenoid units formed from mevalonate, right? Now 5 carbon units are formed. You have to condense 6 5 carbon units, that is 30 carbons is going to be formed, and that particular 30 carbon compound is squalene. So how do you form squalene? So here you had the isopentenyl diphosphate and dimethylallyl diphosphate, right? So 5 carbon, you form a 10 carbon compound which is geranyl diphosphate, 10 carbon compound. Then you add one more isopentenyl diphosphate and you form a 15 carbon compound which is farnesyl diphosphate. Geranyl diphosphate, then you have farnesyl diphosphate. So 15 carbon compound. Now two 15 carbon compounds join together to form your squalene. So one more farnesyl diphosphate will be added and the enzyme squalene synthase will synthesize squalene, which is a 30 carbon compound. Now this does not have the steroid ring. Now you have to form the steroid ring. That is what happens in the next step. That is cyclization of squalene to form the first sterol, which is lanosterol. So if you see here, the enzyme is squalene epoxidase, which forms squalene epoxide, and then it is acted upon by the cyclase to form the first sterol ring. It is a 30 carbon compound and that is lanosterol. This is the first sterol ring formed. Now, through a series of steps from lanosterol, you synthesize cholesterol molecule, which is a 27 carbon compound. Lanosterol forms many series of intermediates like zymosterol, desmosterol, and ultimately it forms cholesterol, which is 27 carbons. So just to summarize all the steps, we started from acetyl-CoA, synthesized mevalonate first. From mevalonate, we formed isoprenoid units. From isoprenoid units, we formed squalene. From squalene, we formed lanosterol. And from lanosterol, we formed cholesterol. So this summarizes the entire steps in the synthesis of cholesterol. Now, once this cholesterol is going to be formed, what happens to its metabolic fate? What happens to it? What are the pathways it is going to follow? So, here if you see, cholesterol molecule can go into the synthesis of its special products such as bile acids and steroid hormones, and in the skin it can lead to the synthesis of vitamin D also. In addition, it can get added to a fatty acid, or a fatty acid can get added to this cholesterol molecule and the third OH, and it forms a cholesterol ester. And also it can be incorporated into membranes. We have already seen the function of cholesterol where it gets incorporated into the membranes and it is responsible to maintain the fluidity of the membrane. Now let us see the clinical significance. You all know, right? High cholesterol levels are going to be very dangerous to the human body. So why? Because this cholesterol can get deposited in the various blood vessels and lead to atherosclerosis, and that can lead to blockage of the blood supply to vital organs, and it is highly prone to the development of cardiovascular disease. Cardiovascular disease and atherosclerosis are the most relevant clinical manifestations of excess cholesterol levels. And in addition, we have certain lipid lowering drugs which can be used in the treatment of dyslipidemias, which is currently prevailing as a epidemic worldwide. Okay, so these lipid lowering drugs, we have already seen one, remember? Yes, HMG CoA reductase inhibitor, that is statins. That is the most commonly prescribed lipid lowering drug. We have various statins, atorvastatin, simvastatin, lovastatin, pravastatin, and there are many statins there. Then we have something known as ezetimibe. Ezetimibe is a cholesterol absorption inhibitor. It acts on the Niemann-Pick type C protein. This is a very commonly asked MCQ. Okay, ezetimibe acts by inhibiting cholesterol absorption, and the particular protein on which it acts is Niemann-Pick type C protein. Then we have the various fibrates. You know, fibrates can lead to increased clearance of LDL cholesterol. LDL cholesterol is bad for health. So when you clear it at a faster pace, it is going to lower your lipid levels. And in addition, these fibrates can also lower your triglyceride levels. And finally, not commonly prescribed nowadays, we have the bile acid sequestrants, and the most common example is cholestyramine.